Good morning, friends. Today we are going to start with the third topic from fluid physiology, that is ascent of SAP. In last topic, we have studied absorption of water mechanism. So, water absorption and ascent of SAP, these both phenomena they are related with water. In water absorption, we study how water enters inside the xylem of the root from soil solution. But in ascent of SAP, which is also another important physiological phenomena, it is concerned with transport of that absorbed water from the root to the leaf of the plant. So to ascend means upward movement of sap, that is from root xylem to the leaf xylem of the plant. That process what we call ascent of sap. Sap means it is nothing but the mixture of water and different mineral salts dissolved in that soil water. So soil water, it is not pure water. It is the mixture of water plus different types of salts dissolved in it. So this mixture we call it as a sap. So ascent of sap means movement of water molecules from the xylem of the root to the xylem of the leaf. When the plant absorbs the water from the soil, along with water, the plant also absorb many mineral salts. So that pure water is not there. Water plus salts they are absorbed by the roots. So it is called as sap. And such sap from the xylem of the root, how it moves towards the xylem of the leaf, where it is utilized for photosynthesis and other phenomena. That process that we are going to study in this topic, ascent of sap. Then how we can define this phenomena ascent of sap? Now, it is the movement of water through vascular system, that is xylem, from root xylem to the leaf xylem. It is called ascent of sap. Sap in the cell, we call it as a cell sap, and sap which is present in the xylem, it is called xylem sap. Look at this figure here. Root hairs are shown in the figure, which are directly in contact with the soil solution. And in last chapter we have studied how the water enters from the root hair to the epiblema, then cortical cells, endodermis, pericycle, and finally how it reaches to the xylem of the root. Okay, so this central part, it represents the xylem of the root. Then once the water enters into the xylem of the root, the process of water absorption is completed. Now the next phenomenon, that is ascent of sap, starts from water which enter into the xylem of the root and how it reaches to the xylem of the leaf, that we are going to study in this topic. Like that, from xylem of the root, the water is conducted by the xylem tissue to the xylem of the stem. From stem xylem, it will be conducted towards the xylem of the leaf, and from leaf xylem, that water will be utilized, part of water will be utilized for photosynthesis, rest of the water will be evaporated through the stomatal opening pores of the leaf surface, and it will be given in the form of vapors to the atmosphere. So this is what we see in this figure. Now same is shown in this another flow sheet. See from the base, soil solution where the sap is present, that is mixture of water plus salts. Then that sap enters through the root hair, through the epiblema, then cortical cells of the root, endodermis, pericycle, and finally xylem of the root. Then from xylem of the root, it enters to the xylem of the stem, from stem to the leaves, shoot apex, then finally leaves, and as shown in the figure, through the stomatal opening pores, the water is evaporated into the atmospheric air, what we call the phenomena of transpiration, that we'll study in the next topic, no doubt. So this is a very simple phenomena shown to explain the phenomena of ascent of sap. To prove this ascent of sap, how it takes place, what are the experimental evidences to explain the path of water in the plant body. We know the conducting tissue consisting of xylem and phloem. Then which tissue is responsible for the ascent of sap? That question arises. Whether phloem is responsible for ascent of sap or xylem is responsible for ascent of sap. To solve this problem, many plant physiologists put forth many views to explain the path of the water. First experiment called as balsam twig experiment. In this experiment, a balsam plant, during this rainy season that balsam plant twig grows very vigorously in the hilly areas. So this balsam twig, which is having the pink colored twigs, leaves, etc. These twigs were taken for this experiment, and that is called balsam twig experiment. These two balsam twigs were put into the conical flask containing eosin, one conical flask containing eosin solution and another conical flask with pure water. The lower end of that balsam twig was slanting cut taken, and then these twigs were put into that conical flask containing eosin and water solution. It is observed that in the first twig, which was dipped into the eosin solution, it is observed that the central xylem tissue of that balsam tube got red colored, that is color of eosin solution, while the phloem tissue, it was not taking that red color. When we take the transverse section, then it was observed that red color of eosin, it was observed in the xylem tissue. So what it indicated, this experiment? These experiments indicate that xylem tissue is responsible for ascent of sap. That is the eosin solution, for example, it is a mixture of water, that is what we say sap, and that sap enters through the xylem, and so the xylem tissue of the balsam tube continuously get colored red color. So this experiment proves that xylem tissue is responsible for ascent of sap. Second experiment to prove the path of the water as a xylem is called ringing experiment. In this experiment, two twigs are taken. Their lower transfer cut is given for two twigs. Then bark of one twig, that is outside bark, that is bark containing phloem tissue. Bark of one twig is removed and it is kept into the water. And the second twig, its central part, central core or xylem tissue, for example, that xylem tissue was removed and that twig was put into the water. After half an hour, it is observed that the first twig, in which only bark was removed, that twig was observed to be fresh, while the second twig, of which the central xylem tissue was removed, it was observed that this twig get wilted. Why this happened? Because as we have removed the xylem tissue, so the water from that medium, it could not get absorbed because of absence of xylem, so that we get wilted, leaves get wilted. While in the first week, the central part, that is xylem, was kept intact, and only external phloem tissue was removed in the form of ring. So as xylem was intact, so water was absorbed by that xylem, so the twig was fresh. So this is what this ringing experiment proved. Xylem tissue is responsible for the conduction of water. And third experiment, called as paraffin wax experiment. In this experiment, one twig is cut at the lower side and put into molten paraffin solution. What happened? In molten paraffin solution, then fresh cut is made at the twig base and kept in aniline solution. And another twig was selected, which we call it as a control twig. It was also placed in water. The shoot placed in paraffin wax started wilting of leaves, while the control shoot, it shows leaves fresh. It means when the twig was put into molten paraffin wax, what happened? That paraffin wax was absorbed, liquid paraffin was absorbed by the xylem tissue, and due to absorption, what happened? The xylem tissue was blocked. The central lumen of xylem was blocked because of paraffin, and that paraffin later on gets solidified. And such a twig, when we again make a transverse cut and put into the water, due to the blockage of xylem tissue, that twig would not absorb water, so the leaves of such a twig get wilted. So, what it indicates, due to the blockage of xylem tissue by paraffin, xylem tissue was blocked, so the water was not conducted and it was responsible for wilting of the twig. Thus, these experiments prove that water is transported through the xylem tissue perfectly, not through the phloem.